Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today we're talking about topic 3.1, enzyme structure. So what will we learn today? For today's lesson, we're going to look at what is the role of enzymes, how is enzyme structure related to enzyme function, what determines whether a substrate can interact with an enzyme, and finally, what happens to an enzyme after the completion of a reaction. So let's dive in. Enzymes are biological catalysts that speed up biochemical reactions. Most enzymes are proteins, so they have a tertiary shape that must be maintained for functionality. They have a region called an active site. So if you take a look at the picture, you'll notice two different enzymes, two different active sites. A molecule that can interact with an enzyme is called a substrate. Enzymes have an active site, specifically that interacts with substrates. That active site has a unique shape and size. It can have a chemical charge or charges or not. The physical and chemical properties of the substrate must be compatible to the active site. And then finally, slight changes can occur in the active site to align with the substrate. Enzyme names often indicate the substrate or chemical reaction involved. These names often end in ASE. For an example, sucrase. Sucrase is an enzyme that digests sucrose. Enzymes are reusable. They are not chemically changed by the reaction. Cells typically maintain a specific enzyme concentration. And finally, enzymes can facilitate synthesis or digestion reactions. So that is shown in the picture on your screen. So let's practice. For today's practice, the skill focus is concept explanation. So you're going to explain biological concepts and/or processes. So let's take a look at the question. What I would like you to do for your question is I would like you to pause your video. First, read the prompt. Look for the bold-faced word, that is the task word, and then ask yourself, what are you being asked to do? Then reread the prompt and formulate your answer. Once you have your answer, push play again. Welcome back. The question asked you to look at the study of the rate at which salivary amylase hydrolyzes amylose into glucose and best explain the chemical interaction between amylose and salivary amylase. Your answer should have been D. Amylase active sites have similar chemical properties to amylose, allowing glucose products to form. It best explains the chemical interaction between amylose and salivary amylase. Amylase has a polar active site, and amylose is a polar carbohydrate polymer, so they're compatible. So what should you take away from this lesson? First, enzymes speed up chemical reactions by lowering activation energy requirements. The structural characteristics of an enzyme makes the enzyme very reaction specific. The shape and charge of the substrate must be compatible with the active site of an enzyme for a reaction to occur. And finally, enzymes are not consumed by the reaction. Enzymes are reused. Well, I hope today's video was helpful. Until next time, thank you.